let's talk about gram-negative curve bacilli. Okay, gram-negative curve bacilli, which are oxidase positive. There are only 3 organisms. Number 1 is they grow in 42 degrees Celsius. So answer will be Campylobacter jejuni. They show darting motility and they grow in scleromedium, and they cause 2 diseases: Guillain-Barré syndrome and Reiter's disease. Fine. Okay, Guillain-Barré syndrome most common is most common. It's autoimmune inflammatory demyelinating polyneuropathy. There will be ascending paralysis. Okay, from down to upward, and it's a bilateral symmetrical weakness. Okay, and weakness also from distal to proximal. So first clinical picture will be rubbery leg. Okay, time taken to reach maximum weakness is always less than 4 weeks. And here will be areflexia, and bilateral 7 will be damaged, but bladder are spare. Fine. Now gram-negative curve bacilli, which is oxidase positive and grow in alkaline media. So answer will be Vibrio cholera. For Vibrio cholera, transport media is Venkat-Raman Ramakrishnan medium, Carey Blair media, and autoclave seawater. Enrichment media is basically contain of peptone water. It's of 2 types, alkaline or Monsu torochlor tryptocase telluride. For selective media, it's a bile salt agar, thiosulfate citrate, bile salt agar, or gelatin, Monsu torochlor tryptocase telluride. Okay, third for Vibrio, okay, listen, and Vibrio parahemolyticus, it's found in a sea fish or shellfish. So they typically give a history of seafood poisoning, and they will ask which Vibrio is associated. It's basically Vibrio parahemolyticus. Vibrio vulnificus, they basically penetrate gut and enter the blood and direct causes sepsis. When they penetrate the gut, okay, there will be no any GIT manifestation. For example, abdominal pain. Okay, they do not cause GIT manifestation. They directly enter the blood and causes septicemia. So gram-negative curve bacilli, which is produce urea, is Helicobacter pylori. It also shows scleromedia. Okay, it can be grown in a scleromedia like Campylobacter jejuni. Now let's talk about Vibrio. Okay, Vibrio, it can be Vibrio parahemolyticus and Vibrio cholera. Vibrio parahemolyticus is a capsulated. Vibrio cholera is non-capsulated except Bengal strain. Vibrio parahemolyticus are give a safety epinephrine, a bipolar staining we can say, but cholera no. In thiosulfate citrate bile salt, the Vibrio parahemolyticus is a green color colony. Cholera will be yellow color. If you look at flagella, in solid media, Vibrio parahemolyticus so peritrichous flagella. In liquid media, polar flagella. But cholera, it will be only polar flagella. Okay. Salt loving halophile, it's only a Vibrio parahemolyticus. Vibrio cholera is not a salt loving. If you talk about a phenomena, so Vibrio parahemolyticus has 2 strains. That is found in environment, that is in soil, and other is found inside our human body. So those strains which are found in human body, they cause hemolysis on a Vagat soma agar. There's a culture called agar media. Its name is Vagat soma agar. So if we obtain the strain from human being, that causes hemolysis on Vagat soma agar. Okay, and this phenomena is called a Kanagawa phenomena. Fine. Vibrio cholera do not causes hemolysis, they causes hemodigestion. Okay, hemodigestion is basically that bacteria will produce some toxin. That toxin will destroy the RBC. That's called hemodigestion. Okay, so and both Vibrio parahemolyticus and Vibrio cholera, both of them show string test positive. Both of them show string test positive. Now let's talk about gram. Okay, there's a fish I told you that Vibrio parahemolyticus is associated with shellfish, yeah, seafood poisoning. It's also one more bacteria, Pseudomonas. So fish poisoning associated with Pseudomonas, especially those fish known as scombroid or mackerel fish, even non-scombroid fish that has not been refrigerated. That fish are also associated with the Pseudomonas fish food poisoning, seafood poisoning. Basically they are toxin mediated, which is heat stable. Okay, and this fish poisoning associated with Pseudomonas have incubation period, or we can say within 15 to 90 minutes of the ingestion. Okay, and there will be tingling of lips, abdominal discomfort. Okay, once the patient started to eat, the first food will come in contact with lips, so there will be tingling of lips, then it will reaches the abdomen, so abdominal pain, discomfort, then it will absorb, enter the blood, so there will be flushing, and finally deposit in the skin, so it causes a pruritus. And it is what is the treatment for this fish poisoning associated with Pseudomonas? Is self-limiting. Just within 8 to 12 hours the symptom resolves by its own. Now gram-negative bacilli, which are causing not oxidative, here it's a gram-negative curved bacilli. Now only simple gram-negative bacilli. So see, they causes lactose fermentation or Meconic agar. If yes, so see whether they are doing fast fermentation or slow. If they are doing fast fermentation, answer will be E. coli, Klebsiella, Enterobacter. If it is going slow, so answer will be Citrobacter, Serratia. This Serratia produce a red pigment known as prodigiocin. Okay, and this is a red color. So this come in the cough, and it mimics hemoptysis. So Serratia infection patient so pseudohemoptysis. Now E. coli, I say that the E. coli causes lactose fermentation in Meconic agar, but there's one specific strain, enterohemorrhagic E. coli O157H7. They can be differentiated all other type of E. coli. Just add sorbitol on this Meconic agar. Okay, generally lactose fermentation are seen in Meconic agar. So all the E. coli will show lactose fermentation of Meconic agar, but if you want to differentiate enterohemorrhagic E. coli from all other type of E. coli, just add sorbitol. Okay, so sorbitol Meconic agar differentiate enterohemorrhagic E. coli O157 from other E. coli if they do not show lactose fermentation Meconic agar. So see, this organism causing oxidase positive or negative. It is positive, so the answer will be Pseudomonadaceae family. For example, S10 Notrophomonas maltophylla, Burkholderia, Pseudomonas aeruginosa. Okay, there's a disease called ecthyma. If it is ecthyma gangrenosum, it's caused by Pseudomonas. If it is ecthyma contagiosum, it's called a parapox virus. Okay, if suppose these are oxidase negative, next step C, whether they are producing hydrogen sulfide on TCI agar. If yes, the answer will be Salmonella and Proteus. If they do not produce, the answer will be Shigella and Yersinia, which basically Yersinia mimics pseudoappendicitis. Now see gram-negative organism. Okay, if they are diplococci, so answer will be diplococci. So this diplococci is stick aerobic. Okay, so it can be meningitis, Neisseria meningitidis, and Neisseria gonorrhea. Example of Neisseria gonorrhea is Moraxella. So gram-negative organism which are diplococci, they are strict aerobic. Okay, so answer will be Neisseria meningitidis and Neisseria gonorrhea. Neisseria gonorrhea example is Moraxella. Gram-negative which are Coccobacilli, which are Haemophilus influenza, Bordetella pertussis, and Brucella. After that, Acinetobacter baumannii, Francisella tularensis, Pasturella. So this is a chart of a gram-negative. First I discuss gram-negative curved bacilli which are oxidase positive, then gram-negative bacilli on the basis of lactose fermentation, and finally gram-negative which are diplococci and Coccobacilli. So we have classified gram-positive and gram-negative organism.